I'm Mark Mers, 2024 president of the Appalachian Highlands chapter of Wild Ones. Whether you're joining us in person or online through our Zoom link, our organization is proud to sponsor this event. We're hoping you'll look us up online at wildones.org, where you'll discover that our mission is to promote environmentally sound landscaping practices to preserve biodiversity through the preservation, restoration, and establishment of native plant communities. Ben Casteel's talk tonight on vegetative plant propagation certainly advances our mission, and we thank him heartily for helping us gain the skills we need to restore natives to our home landscapes as we work to shrink our lawns. There's a lot I could tell you about Ben. Here at Virginia Highlands Community College in Abingdon, Virginia, Ben is program coordinator for the Agriculture Management Certificate and Horticulture Productions. He represents the faculty for the VHCC Board of Directors, and he manages the Treadwell Jones greenhouse from which we're broadcasting. And he coordinated the effort to get VHCC recognized as a Tree Campus USA. Outside VHCC, Ben is engaged with the public schools. He promotes seed exchange programs. He teaches community courses in gardening and is a board member for Appalachian Sustainable Development. His personal life involves family, gardening, and playing mandolin in a bluegrass band. And finally, he was an AT through hiker in 2006. So welcome, Ben Casteel. And if there's anything else you feel like you need to say, then have at it. And the camera's yours.
Thank you so much, Mark. Thank you all for doing what you guys do. Wouldn't be here without you tonight, obviously. But also, I mean, I think Wild Ones is just an excellent group, and you all are doing wonderful things. So, couple of housekeeping things. If y'all are joining us from in person here, bathrooms just out this door, and water fountain. And yeah, if you need to get up and do anything, feel free at any point. If you wouldn't mind, hold your questions till the end. That way we can kind of get through the presentation, and we'll do a little bit of Q&A at the very end. I've got some plant props I'll share with you guys, and I'll try to get them kind of on the screen maybe as we talk about them. And you can kind of look at some of the propagation that we've done here on campus through the process. If you guys are joining us from Zoom, I've muted everybody, but you are allowed to unmute yourselves. If you have a question, probably pop it in the chat whenever it comes to mind. And again, we'll hit those questions up at the very end, and maybe keep yourself muted until then. And I'm going to share my screen here in just a second. This recording will be available later on through Wild Ones. I'll send the link out, and then they'll be sharing it to the YouTube channel, and whoever else would like to watch this later on or refer back to it. So we've got a lot to cover in our time here tonight, so I'm going to go ahead and get started. Guess I should probably turn the screen on for you guys here, huh? Okay, well, so last time we were together, we talked about native plant propagation from the seed side. So tonight we're going to talk about native plant propagation from the vegetative side. So sexual versus asexual propagation. And I wanted to just kind of start off kind of considering first something just inspirational, right? So, and nature is the inspiration for us here. And in your kind of native plant landscapes, I think it's important to rely on, the natural world, the native habitats. And that's one thing that we always want to go back to is using, what is growing wild in this particular aspect on a north slope, on a south slope, and trying to kind of emulate that. Our ecosystems are a little bit different. I think about, kind of the permaculture design aspect of like the zone 5. You guys familiar with that? Like they have zonation. Think of zone 5 as a wilderness area, really, something that we don't really touch. We're not looking at that area in terms of our propagation efforts. We're really looking here at the other zones and kind of bringing things into our personal spaces. Most of what we look at for propagation will be looking at maybe taking things from like the zone 4 areas that we don't visit as often, or from private land areas, and bringing them into our gardens. But this image, it was something that sticks in my mind from my time hiking the Art Loeb Trail in North Carolina. Has anybody ever been on that one? Beautiful area, Western North Carolina, the cold mountain region. And you hike up to the Blue Ridge Parkway, and it's this long, steep incline, and you're in woods, right? You're in the forest. And as you pop out from the forest onto the Blue Ridge Parkway, all of a sudden you've got this kind of ecotone, right? Where this grassland sort of enters the forest. And on that edge, I see this beautiful bloom of bee balm kind of mixing in with some pale-eyed coneflower, or however you wish to call it. So many different common names, right, for these plants. Shall we say Monarda didyma and or didyma and Rudbeckia laciniata. We'll take the scientific approach for most of our discussion tonight, because the scientific names are the best to go by, right? When we're looking in the literature, that's where we'll find the best information. However, if you need the common names, just poke me there and just say, hey, what's the common name on that? So yeah, pale-eyed, or let's see, green-eyed coneflower, they call it sometimes, but it's actually a Rudbeckia, closer to black-eyed Susan or brown-eyed Susan. And just that color contrast, just the lime green of the foliage with the bright scarlet of the bee balm and that bright yellow of the Rudbeckia, just something that I've always wanted to recreate. And so finding that inspiration and then coming back and saying, I want to make that in my habitat. We are a keystone species in our own, and well, really in all ecosystems, but especially the wildlife habitat that we create around us. So we kind of, in many ways, dictate what will grow and what will survive and persist. And I think that it's always best if we take those cues from nature. All right, well, let's get started here talking about the asexual side, which is really also plant cloning. And there are some things about that. A little bit of review first, that before we get into asexual propagation. First, what is plant propagation? Well, that's obvious. It's the reproduction or increasing in the number of plants. And we do that via the two main means, either the sexual, which we talked about last time, requiring flowers of the opposite sex. We really want to look at that, the genetics when we think about that. We also talked a lot last time about stratification and scarification requirements to get seeds to germinate. Tonight we'll talk about the asexual propagation aspect, which only requires one plant or just some plant tissue. Again, this is cloning. And there are many different techniques to get plants to produce asexually. And these techniques are often particular to the family or maybe even the genus of plants. So we'll talk about some techniques that can be applied broadly, but also some that work better to specific genera or species, families. Okay, so on the sexual propagation side, we are reproducing plants from seeds resulting from fertilization. So I think I kept bringing up milkweed last time because we think of it as being maybe a mascot plant in many ways because of the knowledge that we have from how it affects pollinators, but there are so many more pollinators than the monarch butterfly that are important to consider, and many more plants than just the common milkweed, Asclepias syriaca, that really need to be propagated and could be fostered in our own gardens. So on the sexual propagation side, it has some advantages. It's fairly a quick way to get many plants started at once if you have a lot of seeds. It's fairly easy, right? I mean, talk about using the milk jug method, making like a little miniature greenhouse. That seems to do very well for some plants that require stratification. I've been watching Gail's Facebook. She's got, if you need some plants, holler at Gail, right? She's got all kinds of seeds that she started through that method. So it can be fairly easy too. And it's economical, right? You can upcycle some plastic containers and get a vessel that can easily grow you a lot of plants. However, there's some downsides to sexual propagation, right? Probably the biggest one, some plants, especially hybrids, which hybrids do occur in nature too, they do not reproduce true to the parent plant that they came from. So in order to get, a reproduction of the parent plant, a clone, we really can't undergo or save the seed from it. And of course, some plants are extremely difficult to propagate from seeds. I mentioned one last time, and kind of a project that we're working on with the showy lady slipper, Cypripedium reginae. The seeds are extremely, well, they require a mycorrhizal symbiotic relationship. And if you don't have that mycorrhiza, they won't grow. So you kind of have to use, in this way, sort of a combination of sexual and asexual techniques. And I'll kind of refer back to that here closer to the end of our talk tonight. All right, so advantages of asexual propagation, kind of the opposite of what we just talked about there. One, we can get larger plants to mature in a shorter time in some cases, not in all. That's not like a blanket statement, but we can if done properly. Some plants, of course, do not produce viable seeds and therefore would require us to propagate them asexually using the vegetative tissue. We can also salvage genetics through asexual propagation. And a good example of that is when a plant gets overgrown in your garden, starts to crowd other plants. Sometimes through asexual propagation, we can cause the parent plant to recover, or we can save something. Maybe it's ridden with pests, right? Or it has a disease. Sometimes we can actually, through asexual propagation, clean that tissue and salvage those genetics. And of course, my animations are going to be fighting with me all night. The downsides of asexual propagation, before we get into it, is one, some of the methods do require special equipment and some, a particular skill set to be able to do them. A lot of the plants that we're going to work with, in the wild ones arena are not well documented in terms of the asexual propagation. So we don't have good guidelines to go by in some cases. What I've found is a good way to go about that, if we're going to try to asexually propagate them, is to look at the closest relative and maybe try to apply similar techniques to that. Also, if we don't do it right, we can damage the plants too. And can't get quite as many plants through asexual propagation started at once as we can from seed. Also, now this is something we probably aren't as concerned about in the wild ones arena, but there are some nativars that do have plant patents, and technically there are two levels of plant patents. One where you can't propagate them, period. Who's checking on that? But the other one, you can't propagate and then sell them. So that's the biggest plant patent issue. So a few guidelines before we get into talking about the different types of asexual propagation. And it's similar to guidelines that we would use if we were, say just foraging too, right? So first, use the technique for asexual propagation that is appropriate to that plant. Otherwise, we would just be wasting the plant and its tissues. We'll talk more about that and unfold what those appropriate techniques look like for the different genera and species. Also, think about that. What technique is appropriate to the site itself? Are you in more like a wetland ecosystem where you could maybe alter the hydrology by, digging into plants? Are you in a private land, versus, well, you shouldn't be doing any of this on public land unless you have a permit. Also, whenever it comes to asexual propagation, just as with foraging, we want to harvest a minimal amount. So something that's not going to damage the ability for the population to continue to reproduce on its own. I mentioned the private land thing already, but you can't say it enough. There's tons of private land out there with lots of great natives growing. And, that is, more acceptable, shall we say. We can't do it on public land without a permit, right? So you can get permits. Forest Service, for instance, will allow, propagation of particular plants or gathering of plants if you go through the permitting process, which I think some of us could probably talk a little bit more about that, right? Or are they easy? No, it's a long documentation process. However, some plants, like maybe if you're foraging for them, right? You're allowed to forage, like for ramps, for instance. It's like there's an allowance for foraging for them. You could also be propagating them while you're foraging, right? So
Change your mind.
yeah, maybe some loopholes here and there. All right. Well, anyways, those are the main rules. Can you guys think of anything else that we would want to include here? Be mindful, right? Also, I guess one thing I should include here, know the plant that you're working with, right? Some plants are poisonous. Some plants can have some negative effects. So we want to be aware of all the properties the plants have and, whether they're rare or not, and anything that we could do that damages them or damages us. All right, so in short, asexual propagation is the use of the vegetative growing parts other than the sexual growing parts. And think of all plants as having that life cycle. They're going to go through a vegetative phase before, whoops, no, this is going ahead of me here. They go through a vegetative phase before they go into their reproductive phase. And in the vegetative phase, they are easier to propagate and more likely to develop roots. So here are the types and techniques, and I'm going to say these kind of in this order because I think that this order is going to be kind of the least potentially damaging. Cuttings, you're not really damaging the plant tissue if you're doing it carefully. Layering, a little bit more potentially damaging. Grafting and budding, kind of the same deal, which has kind of limited use. Tissue culture is not damaging at all, but it requires like some extra steps and some advanced techniques to be able to do well. And then finally, division and separation, which is an excellent technique to use. However, it requires you to dig the plant parts. So therefore, you're more likely to damage other plants around them. So I put that at the end. It's probably, in our own gardens, what we would use first, especially if we're looking at rejuvenating parent plants. But we think of it as being more potential to damage a population. All right, so let's start with cuttings. And with cuttings, we're taking that vegetative tissue and we're really trying to produce roots. So we're rooting cuttings. There's three main types of cuttings, depending on the tissue itself. The first would be stem cuttings. And a great example of a plant that's, or a plant family, I should say, that is easily started from stem cuttings. I'm sorry, guys, my animations they have a mind of their own, I think, or maybe they're timed somehow. I need to turn this timer off. Anyway, bee balm right here and the mint family are great for stem cuttings. A lot of our succulent plants, which this is an image of our native succulent, sedum, one of our native succulents, Sedum tornatum, the wild stone crop. It can be grown from leaf cuttings. So in this case, a leaf tissue can become a brand new plant. And then, of course, there are some plants that actually grow from the roots themselves. And rather than producing new roots, we're trying to produce new shoots. And this is what a root cutting could look like. This is a persimmon. So persimmon is a native tree that the absolute best asexual propagation technique for it is a root cutting. Who would have thought, right? You can dig up a section of root. Often plants that tend to develop suckers away from the parents are easily, will more easily start from root cuttings. All right. For cutting, some tips and techniques for cuttings. We have one, hormones. So plants have hormones too, right? We need to have a source of the particular hormones that are signaling the plant tissues to develop roots. So in asexual propagation, we are usually looking at trying to develop new tissue from what we call the meristem, right? The meristem is where cells are actively dividing, but also differentiating. So as a cell gets a cue from a hormone, it could become, instead of a stem cell, literally and figuratively, it could become a leaf cell or a flower cell. You know, switching it in by the hormone induces it to become that other tissue. And so auxin is the class of hormones that we mostly look at for producing roots. And it's actually, if you really want to get scientific, see some science lovers out here. So it's actually a ratio of auxin to cytokinin, which cytokinin is more producing shoots. And we have to have that right ratio. But in general, we want to add auxin. Some plants already have it in their tissue. Those are the ones that root very easily. In this image, we have a rooting gel. And I recommend the gel over the powder personally because it's more expensive, but it goes further, it sticks to it better, and it seems to produce roots faster. So getting the gel in bulk, maybe putting it in a shot glass for when you're dipping it. That is a great way to get that source of auxin, that hormone right there. IBA is the synthetic version of it. I think it's indole butyric acid or something like that. Don't quote me on that. But it modeled, the synthesized version modeled after a natural hormone that occurs in willow plants, willow family plants. So you could just make a willow tea as well. So a weeping willow will work. Corkscrew willow will work. You could use native willows too. So black willow, widespread native willow can also, all you really need to do is stick those branches in a vase of water, watch them produce roots. That tea that is produced will actually have that auxin, the natural form of it, IAA, indole butyric acid, I'm going to say, even though I think that's incorrect. But IAA is the naturally occurring form of it. That's one way to get that hormone. So you can buy the powders, you can buy the gels, or you can make your own. And if we really want to be native purists, then maybe we want to get that native hormone from a native willow. There are other sources out there. That's just the most common. So willow tea, great source of hormone to get your cuttings to root. Second thing that we often need to have especially for woody plants. Now, herbaceous plants tend to root quicker, and because of that, you often don't have as much of an issue with fungal infections. But often your woody plants can take a long time. Some plants up to a year before they'll produce roots. Like the Calmia genus, for instance, mountain laurels. You can get them to root, but it takes forever. So in that case, we do need to provide some fungal protection. And one thing that you need to look for fungal protection. I mean, you could go the fungicide route, of course, but there are some kind of less, shall we say, less costly routes. Hydrogen peroxide, I think, is one of the best. Good old H2O2, which they got it on sale at Kroger right now for $1. You get a quart of hydrogen peroxide for $1, and that will last several seasons. So hydrogen peroxide is great. Not only is it great for fungal protection, but it also will oxygenate as well. So you can add it to media, especially if you start to see like algal growth. You start seeing anything green in your soil or your substrate, get hydrogen peroxide in there. That added oxygen will kind of help to alleviate where algae has kind of depleted the oxygen in your substrate. When it comes to rooting, we also need to look at our substrate itself. Also wanted to mention that on the fungal protection front, I see this as a combination a lot, cinnamon and honey. Cinnamon is a natural antifungal. Honey is naturally antiseptic. So like combining the two, and also honey will help to make the cinnamon stick. So it's like you can get a little bit of protection there. It can be effective to help get some cuttings to root. Last thing is the media itself, the substrate that we're rooting in, which you could root cuttings in water alone. I often find that it takes longer and requires more attention to root in water than it does in a soilless media. And generally, we want to use soilless media. And what I mean by soilless media is a mix, like what we have in these containers, of usually peat, bark, perlite. These are the things that are often used in the soilless media. Your Pro Mix is a good example of it. A bag of potting soil, is soilless media. Usually we don't want to use garden soil, mostly because there can be, lots of stuff in there. Could be pests, could be fungus in our garden soil. Also, it's just really heavy, and you don't get as much of a good contact, good, shall we say, oxygenation in that kind of root zone when we're trying to produce roots. So a sterile media is a good way to go. It doesn't have to be sterilized necessarily. I think getting potting soil has been sterilized at some point. So it makes a good substrate to use for rooting cuttings. But there's many different routes to go there. I just wanted to mention that typically garden soil is not the route to go for rooting cuttings. It doesn't mean you can't. You could absolutely stick cuttings, right into your garden soil and have some success. It's just likely that their success rate is going to be decreased due to the presence of potentially fungus and other pests. So that's the third thing I would mention is trying to get sterile media. You could reuse potting soil too, but you definitely do want to sterilize it before you reuse it. Hydrogen peroxide would be a good way to sterilize it. I hear of some people sticking it in the microwave or the oven too, which could be effective. All right, so again, hormones, fungal protection, and sterile media are big cutting techniques and tips. Also, something I didn't mention here is select vegetative tissue, right? So think about the energy flow in a plant. If it's flowering, it doesn't mean that it won't root, but it means that that plant has put its energy into developing flowers and probably seeds. So I often recommend, if that's all you've got, to try to start cuttings from, to remove the flowers. That way it should put some of that energy back into a vegetative process, kind of start from scratch, if you will. But it's best if you're, for most things, to take cuttings usually during or right after a flush of growth, of vegetative growth, before it's in its reproductive phase. Some things that are easy to root will doesn't matter, but that's probably our best guideline and principle. All right, next, very similar to cuttings, except we're keeping it attached to the plant, is layering. So layering is the rooting of plant parts while they are still attached to the parent. And we'll look at a few different types of layering here, including air layering, trench or mound layering, and compound or serpentine layering. So we'll go through the process for air layering, and it's each of them. Compound and serpentine layering would be for things usually with a flexible vine style of growing. And usually what you're doing with compound is you're burying a part, letting it come up, burying it again. So lots of our native vining plants, our lianas, work really well for that. First, here's just the process of air layering, and there's so many different things that you could use for this. Air layering tends to be very successful because of its attachment to the parent plant, which means it still has a source of water and nutrients, and also the hormones themselves that cause the cells to differentiate and become root cells instead of, say, a stem cell. So for cuttings, I recommend, that technique works better for herbaceous plants. Layering is often better for our woody plants. It can take a while, but it's usually successful. So the big thing is you have to be able to keep it moist. This is probably the most simple form of air layering here. So wrapping a bag, some kind of plastic, a container, right? I've seen people use old soda cans, old plastic bottles. Again, think about upcycling, right? Rather than throwing away, since we can't recycle plastic anymore around here, let's see if we can use it to make new plants. So you could use Ziploc bags. You could use any number of things. They actually make little air layer pods too that you can purchase. But the process is basically, it is going to require some wounding of the plant. And the wounding is going to cause some of the nutrients and the photosynthates that are produced in the leaves, and also some of the hormones to kind of accumulate where it has been wounded. And that can look like a cut right here on the stem. You could kind of cut into it. You could start to girdle part of the bark. You could also just bend it even. I've seen people, especially with more flexible tissues, just kind of give it a bend. That will also cause accumulation. And in our plant propagation class, we start to research all these different ways, and we have students do different cuts and different types of wounding to achieve this. And ultimately, doesn't seem to matter what you do. It's as long as you've done some sort of wounding to the tissue to allow a place for the roots to grow. Now, if it's a plant that has what we call the adventitious roots, those that kind of naturally form on the tissue, you don't need to wound it. But if it's any other type of plant, you really have to at least penetrate the bark. So we're going to wrap something around there, and we're going to fill it again with kind of our sterile substrate, ideally that is very moist. Often we'll mix in a little bit of like willow tea or something to have another additional source of that hormone that will cause it to develop roots. I also advocate for the use of the Terrasorb. You all familiar with that stuff? It's the same stuff they put in diapers, which it's sounds awful, but it's very effective in keeping moisture right around it. Usually, the number one issue we have with layering with students is that it dries out. So you want to try to get it wrapped tightly. Also, something they don't do in this image, but I would recommend doing is probably also wrapping it with, say, like aluminum foil or something, so that you get another barrier from the light. Roots don't typically like to be exposed to light or air. So keeping a good tight covering, and then also preventing it from being constantly irradiated will probably help to keep it moist, but also give the roots a stronger fighting chance there. So using twist ties at the top here, tying it shut, keeping it nice and moist. I like to use something transparent with an opaque covering over it so we can check in on it. Remove that opaque layer, take a look and say, oh, do I have roots growing yet? So layering, air layering can be very effective for plants that otherwise are difficult to root from cuttings. The limitation is you can't get as many off of it. So you're limited kind of by the number of branches. But lots of native woodies will produce roots from layering. Mound or stool layering, typically we top the parent plant and then allow resprouting to begin, and then just mound it up with soil. So rather than doing it on the air and completely covering it, we're just covering it with soil. And as the resprouts will grow, as they develop those shoots, they'll also develop roots along with the shoots. So this is what mound or stool layering could look like just from this illustration. You can see they topped it and then covered it with soil. And then as these new shoots grow, they'll also produce roots. So at the end, we would uncover that mound of soil, snip it at the base below those roots. We have roots, we got shoots, we got a new plant. This is how fruit tree rootstock producers produce the rootstock. So it can also be applied to our native plants. And often I see that this is done in poor landscaping when you get lazy landscapers. They throw the mulch right on the plant gets buried by some mulch right there. It's just like mound layering. You come back, pull some of that mulch off. Hey, I've got some roots. Snip here and boom, I got a new plant. This works well with multi-stemmed shrubs. So like the Physocarpus genus, for instance, 9-bark. A lot of the spirea can grow this way. And I think any multi-stem plant could be grown doing a mound or stool layering. Again, we are limited by the number of plants that we can get, but in Oregon, where they produce most fruit tree rootstock commercially, they will do entire rows of them. They'll cover the whole row with sawdust. They'll snip, they'll come back, they'll cover them up again, and they'll just continue that process and get, hundreds of thousands of rootstock started from just a few rows of trees. Pretty interesting, good way to get some plants from one plant, new plants. All right, so let's talk shift gears. Since we talked about rootstocks, maybe we can talk a little bit about grafting, which I think the grafting application might be a little more limited for what we are doing, but I'll share with you what we use grafting for with native plants. So in general, grafting is connecting of two different plant tissues to grow as one. And it's usually used in mostly with fruit trees, right? Because they don't breed true to type. And we can get some characteristics from the rootstock that could also strengthen the scion. So the scion is the top part that grows and will produce new flowers and fruits. That's the tissue that we want to reproduce. The rootstock is what we're fusing it to, and it's more giving us the root system. Sometimes it bestows Some characteristics such as disease resistance. In the fruit tree world, we use it to dwarf, control the form of a plant, dwarf or semi-dwarf. So here we have an image of budding. That's another form of grafting. The thing with budding is that we're just using one single bud rather than like a whole stick or scion. What I have, this is a pawpaw right here, grafted pawpaw. And can we see a show of hands here of who's eaten pawpaws before? Who likes pawpaws? Hey, that's good. Now, let me ask you this. Have you ever had a bad pawpaw? I've had quite a few bad ones that have like a kind of a bitter aftertaste. And what we're doing here, I mean, really, pawpaws are an example of really a native-are, if you will, because it's a native tree, right? It's a native fruit. But some, ingenuitive growers have found that some have better flavors than others or less of a seed count. So they're not really as much a true cultivated variety as we see with like other fruits, but they can be very important, right? So if you've had a bad pawpaw, and often I say to people that don't like pawpaws, you probably just haven't had a good one yet, right? So there are some kind of bitter off flavors in some of them. You can get around that by cloning those that have good flavors. So these are, this is kind of a project we've been working on here. One of our plant mascots is the pawpaw, and there are actually supposedly over 200 varieties of pawpaws that have been selected by growers out there. We're trying to preserve as many of them as we can. We've got 25 varieties now that we just grafted last year, and we're starting to plant on campus. So this is a grafted pawpaw. I'll pass this around for you guys to check out. This is coming from root stock from just wild pawpaws, or some started from seed. These came from the Department of Forestry in Missouri. That's where our kind of pawpaw guru Woody Walker got them, and he had a bunch left over. So we got them. We potted them on in these deep pots because pawpaw root systems can be extensive, and they don't like to be disturbed that much. So
how long has that been grafted?
That was grafted just in July or August. So that's it got a little bit of a new shoot. It'll probably explode in new growth this year. So some things I've also thought, and I know some of y'all have ties to the Chestnut Foundation. I've kind of mentioned this to Vasily a little bit. If there are root systems of American chestnut out there, could we graft the disease-resistant hybrids to them to reproduce? Maybe. It's a thought, so he's interested, and I think they're going to do some trials this year. The most common grafting technique is pictured here. This is a whip and tongue graft. And so it's not super useful for us to propagate new plants, but let's just say that there's a particular characteristic that we're searching for. That could be a way, and for pawpaws, it's really the only way to clone them. They don't root from cuttings. You could dig up the root suckers, but they usually have a very high failure rate from dug root suckers. So the best way to do it is grow rootstock from seed and then slice a new variety onto it. A little more of an advanced technique, but we kind of went back and counted someone who had never grafted before, a high school student, Ben Elmore. Everybody knows the Elmore family. Ben had just had interest in it, and so we taught him how to do it, and got about 75% success rate. Pretty good for a first-time grafter. So anybody can do it. Seems hard. It's not that hard. Tissue culture and micropropagation. Again, this is something that I think probably has limited application for what we're trying to do. However, it is something that can be very effective. Basically, we're growing plant cells on sterile media. This is typically done in labs, although I did find a good document that suggests you can do it in your kitchen tissue culture. We call them test tube plants. They're produced in vitro. And the thing is that these are used for plants that are very difficult to propagate or to make a whole lot of them. So theoretically, anyway, one cell could become a new plant in tissue culture. Usually we use like what they call explants. So you can kind of see in this image here, it's very often used in orchids. And this is what we're finding we're going to have to do with the showy lady slipper, is to be able to grow it, we need to germinate the seeds on aseptic media. In this media, this kind of gel that we're growing on here, it has like all of the sugars that the fungus would be providing to the developing embryo. So it can be done kind of bridging sexual and asexual propagation, but you could also produce a whole bunch of plants in a very small area using tissue culture. So I'm not going to go too in depth with the process. It's much more scientific. You have to have a sterile area, like a still air box or a laminar flow hood to be able to do it. But it could be something, if you're interested in growing native orchids, for instance, it could be a way to grow them without, damaging a population. All right, finally, talk a little bit here about division and separation, which are very effective. Usually we get like 90% success rate. So division is done through us physically dividing the plants to reproduce. Separation, on the other hand, is more the natural division of kind of specialized plant parts. And this is Liatris. Whoops. Liatris pictured here, blazing star. It has these corms that naturally develop, and we can just kind of pull those corms off. Eventually, the tissue between the cormel, which is kind of like the baby corm, and the corm itself, the crown of the plant, it will eventually naturally kind of separate on its own. So us pulling it apart is more separation. Division would be like cutting the root system, or often we might use a digging fork or a spade to accomplish that. And I've got a few plants up here that are kind of reproduced from both. So I got spiderwort, Triscantia virginiana here, that you can see we went from just one, and just since, this was February that we dug this up, brought it into the greenhouse. It already started to bloom, and it started to produce another offshoot. So what we're seeing is really the crown expanding. This is one that's thicker. This would be a good candidate for division right here. You can, if you start to pull those shoots apart, you can see that really we could get 4 or 5 new starts from that one section of plants. So that would be division, because I had to actually cut the root system to divide it. This is a Jerusalem artichoke, and some of you are gonna be like, keep that away from me. This was produced from separation, and I have this tuber exposed, right? The Jerusalem artichoke is a native plant. This is from a cultivated variety of it, but the tubers will naturally separate from that crown on their own via stolon. And by springtime, that stolon has kind of disappeared, and we just have a separate tuber, which if you don't divide it, it will crowd out. They won't bloom as well, and you'll have a reduction in yield too. So in some cases, separation and division, depending on what technique we're using, can not only produce new plants, but can also help for the population to be more vigorous. And I would refer here, and I save this separation and division for last because it's a little bit more invasive procedure, if you will. But can native plants be invasive? Yes, absolutely, right? So we know that there are some native plants out there that tend to be very aggressive. And when they form really thick colonies, that prevents their ability to sometimes go into the reproductive cycle. I think ramps are actually a pretty good example of that. if you see like really thick stands of ramps, they actually need to be thinned out so that they'll produce seed again. And there's a book out there called Tending the Wild that talks about the Santa Cruz Native American tribe that basically did this with their digging sticks. So as they were harvesting these tuberous plants, they're actually promoting the regrowth of it. And camas was the big one for them. But, Jerusalem artichoke, I would say, was probably one for us because it's here, right? Camas is also here. It's very infrequent, very uncommon. Okay, so here's what it looks like for the spiderworts. That's a big old colony that's like one thick growing. And you'll notice sometimes in your own gardens that when they start to get that thick, they might not bloom anymore. So that's time to divide. A rule of thumb for dividing is that if it blooms in the spring, divide in the fall. If it blooms in the fall, divide in the spring. That way we're kind of sticking with that natural energy flow. The plant parts that we use in division are more like the rhizomes, which is a horizontally growing underground stem. You got to cut it to get a new part. And as long as you have a section that has a root and a bud, you're almost guaranteed to have new plants. Runners and stolons are often also part of that. So our native strawberry, which by the way, was the progenitor of the cultivated strawberry. Strawberry was not a fruit until they crossed to Native American versions. But they produce runners and very easy to propagate via cutting those runners. Stolons are kind of it's a different botanical name, kind of the same principle. Suckers, we might also consider that division too, or root cutting. It's kind of one of those things where at this point it becomes a matter of semantics, right? Is it division? Is it separation? Is it a root cutting? Who cares? It's plant propagation. We're making new plants. That's ultimately at the end of the day, that's what we're trying to do here. So back to our Jerusalem artichoke, involving natural tissues. Anybody wants some Jerusalem artichokes? I got a bunch of tubers that we dug to separate out of the colony because they get crazy. The natural tissues, we're not cutting or breaking. We're just letting that part that comes off of the crown. And kind of the principle of division and separation is that the crown continually expands horizontally as the cells divide. And if we have the specialized plant tissues, it'll have offshoots. If we don't, then sometimes we need to come in here and divide in order to get that crown to not be too crowded. So specialized plant tissues that we're using for separation include things like bulbs, lilies. A lot of our native lilies, you can separate the individual scales and grow a whole new plant just from one scale. Corms like the Beatrice, they make little cormels right on the side of the plant, and eventually they need to come off of there where they'll crowd the parent plants out. Tubers like the Jerusalem artichoke. So these specialized plant tissues are more separation, but again, does it really matter if it's division or separation? Not really. We're just making new plants. Oh, and I guess that concludes our discussion. I'll stop there, and also I wanted to share with you guys a couple others here. Now, this is not a native plant. This is a lipstick plant, but it's just a plug tray. So these are stem cuttings stuck in a sterile media. This potting mix is just straight up pro mix. And often what we would do is cover this with some sort of humidity dome to keep it moist to get it to produce roots. But each one of these plugs could become a new plant, and I think they probably will in this case. So the student that started these dipped them in the hormone before putting them in there, and actually, I would say here in probably a month or so, these will be well rooted, and we can just pop them right out of the plug. So I like to use these plug trays for doing a lot in a very small place. And he did here both stem cuttings as well as leaf cuttings. Like, why not? We'll try to do some leaf cuttings as well. So some plants will reproduce just from a leaf tissue. Some tend to do better from leaf tissue versus stem tissue. Some require the tip of the stem, and that's where most of the hormones are kind of stored. Some don't. So, it's really a fun way to experiment, grow new plants, and typically when we do this, if we have good success, then we have plants to share with others. Also got a dwarf crested iris here that we planted these in our landscape probably a decade ago, and every year we've been going in and dividing a section of rhizome to get some plants. And one thing I would mention about the dwarf crested iris in particular, when is it blooming outdoors? Maybe another month or so. So sometimes you have to be aware of how you're altering the phenology. Same with those spiderworts. I've divided it, brought it into the greenhouse. Boom, it's already blooming. So I'm kind of accelerating some of the processes too by bringing it into a warmer spot, making it think that it's spring. So sometimes you got to time it just right. These tend to do very well though. Either way, it's kind of already bloomed, which is sort of sad. I wanted to have that open flower for you guys to see, but dwarf crested iris, super easy to divide and grow anew. And hey, I think that's about our time here, but I would open it up for questions. Feel free to drop a question in the chat or unmute yourself if you're on Zoom, or anybody have questions, comments, concerns, gripes or grievances?
Could you start the pawpaws with it mounded?
They don't layer well
They
either.
don't layer well.
Yeah. So grafting is like the only way we've reliably found to produce pawpaws other than by seed.
So how do you dilute the hydrogen peroxide? I assume it's diluted.
Oh yeah, you definitely don't want to use it at full strength. I do, like a teaspoon to a quart. It's usually if I'm watering it in. If I'm just using it for, let's say that I do have say a fungus issue or algae issue on a soil surface, I'll just mix it into a spray bottle and I'll use a little bit stronger solution in that case. Probably more like a tablespoon to spray bottle. Rest is water and just kind of spritz it on there. I have used it in full strength, just in a dropper form, and you can see it working. As it starts to kind of fizz and bubble. But that's probably a little caustic for a lot of our plants. So, but yeah, good question. Hydrogen peroxide is super effective as a fungicide to use for lots of issues.
Can she not get those trees off of Woody?
Yeah, she could.
Woody
could buy 'em.
Walker is on the internet, and he will get you those trees.
He'll sell them to you. You could also come. We'll have pawpaw grafting workshops with Woody probably in May if anybody wants to join for that, or June. We haven't had the date set yet, but I'll be sure to let our Wild Ones affiliates know so that we can get that message out. Also wanted to mention a couple other things coming up. April 2nd, we'll be working with the Nature Conservancy to do a river cane harvest. So river cane is our native bamboo, Arundinaria gigantea. There's actually a couple other species we might work with in the future, but right now we're looking at restoring it in order to get stream bank restoration mostly. So we've committed space to do about 500 plants. So that'll be April 2nd, the afternoon of April 2nd. Probably the best way to get plugged in with that would be to join the Nature Conservancy's volunteer list. And then Nick Proctor, who's our community outreach person, he's kind of sending out the information around that. And I could get you guys his email address. I should have brought it with me. It's a collaboration with us, UVA Wise, Department of Conservation and Recreation, and Nature Conservancy. So we'll be harvesting at Clinch River State Park on April 2nd. They have another harvest date, March 26th too. So that's going to be in Lee County, so a little bit further drive. But if anybody's interested in that, I highly recommend getting in touch with Nick at the Nature Conservancy to join for that. But that's a good example of asexual propagation for ecological restoration. And that would be a division of a rhizome. So we'll be basically cutting those rhizomes into sections, and they recommend rooting them in sandwich bags, basically. So it's like just to bring down the cost. So we're gonna have about 500 river cane plants growing in our greenhouse. So if you guys want to come help with that, we'd love to have you. And you probably get some cane to take home too, if you want to raise some cane. Sorry, I had to do it.
Where are they going to replant those then?
So these will go back probably to the sites that they came from, but a lot of them are going to go to natural heritage sites so river cane in particular has been it's a choice browse. So any kind of grazers go for it, and it does not recover from grazing very well. And you might imagine, like, if most of our rivers were probably flanked with river cane most of the way, and most of it's gone now. So stream bank erosion has probably occurred as kind of a 1-2 knockout punch from grazing, first getting rid of the river cane, and then causing the stream banks to be more susceptible to erosion.
Where's the best place to put that?
Somewhere wet, really. Put it anywhere where you would otherwise put bamboo. Don't plant bamboo, plant river cane instead. But yeah, it does like it wet. So it does need to be somewhere where you can either give it water or put it in a place where it gets water naturally. Great for stream bank stabilization though.
And I have a question, but it's really more related to sexual reproduction.
Sure.
And it's about, and it also relates to like the gene pool and a very local population. So I keep hearing arguments between people who say that, a very local population with a restricted gene pool is not as resilient, perhaps to something like climate change or other perturbances. Disturbances. So I mean, what's your thought on that debate? I mean, I think, the perspective probably among us is that if we're using local genotype, then it's probably going to be better suited to our location. But there are others who argue differently.
Yeah, I think that's a great question, and it's a tough one too, tough one to answer. I would say, it's definitely worth the preservation of them either way. And, if we're, the USDA's germplasm repository network, they would call it an accession. So at any gathering event would be an accession. And whether we're storing that, in situ in our own gardens or ex situ in a seed bank somewhere, we at least have it stored. So it could be, if the climate starts to push the southern plants further north, then at least, or the northern plants further north, at least we have a refuge for them. We have a source to reseed them. So that's a great question. And I would say, don't let the naysayers tell you not to do something. I mean, it's still worthwhile to preserve and conserve those gene pools. And I'm a big proponent of provenance, right? So what we find too often is that when we can get native plants, they are not really native here. I'm happy to buy some native plants from like Ernst Seeds or Prairie Moon Nursery, some of these places that have a lot of our native. But again, they're not really native to Southwest Virginia. They're coming from Minnesota or from Pennsylvania. So it's native to North America, Maybe it's native to the mid-Atlantic, but we need people to preserve the genotypes that are here now, because it's not being done nearly as much. Although you all are becoming the stewards of this stuff, and so yes, I think short answer, do it anyway, right? Preserve it, and let's find out for ourselves. Let's grow them and see what happens. We won't know unless we experiment. So don't let them tell you not to do it. Yes, Lavonna.
Do you have a place for those 500 plants?
Oh yeah. So I see 500 plants, like it's going to be like a huge area, but they're putting them in like a section of rhizome into a sandwich bag. So like 500 takes up like a 20 by 20 area maybe. So
What does it do for the wildlife?
oh, it's an excellent habitat for all kinds of songbirds, especially those that nest in riparian areas. It's also, it's just good habitat, period. Lots of insects, lots of, I would imagine probably, our other kind of river mammals, maybe otters. So there's some stuff out there. So it does a lot of good. It is a good solid plant. And if you can make it out, if you can't make it out to that gathering event, I'll save some for you.
Yeah, but that's not the route I'm going. The route I'm going is over there on the river. Plant some of it.
Oh, absolutely. Yeah, it'd be great to plant some on the
Because
river.
there's acres and acres of plant over there.
You had questions?
Yeah, are there efforts, what I'm thinking is are there areas that are being developed where we're losing some of the regional natives where plants could be rescued? Are there efforts directed towards that?
That's a great question. And unfortunately, I'm not aware of any, but I think that could be something that, we should be looking at really, Wild Winds of Appalachian Highlands. Let's form some sort of coalition where they're starting. And we look at real estate developments. Why don't we see if we can get in there? And in that case, that's one time that I wouldn't mind digging. I'm going to say for the most part, we don't want to dig. That's because it's the most invasive procedure. But if it's going to be destroyed otherwise, absolutely. When I went to school in Prescott, in Arizona, Prescott College, they had a local group that was doing that, and they started spanning a pretty good distance, even went down to Phoenix a lot, because Phoenix was just developing so rapidly, and they were destroying the saguaro habitat. And so they were going in and they were having success digging sometimes, maybe 100-year-old saguaro and moving it. So a lot of these things, there it's a little bit different because everything grows so slowly and is very old. But we still have a lot of species here that can take a while. And yeah, absolutely, we should be doing that, and I'd be happy to help. Not exactly sure the best route to go about that, whether it's like just contact like the developing companies, or maybe places where they have to get permits and say, hey, also, why don't you get in touch with this group so they can rescue any plants that you're getting ready to scalp off of this, mountainside.
There is a group in Knoxville called the Native Plant Rescue Squad.
Oh, awesome.
And that is exactly what they do. So that would be a good maybe resource to figure out how they got started. They're housed at the Knoxville Botanical Garden. But yeah, that's what they do. And then they resell to the public.
Great. Yeah. So that's, let's find that contact information. Let's get in touch with them, because that would be, I'm sure you could emulate that process, right? What did they do? It sounds like having a botanical garden is maybe like a sponsor or a host would be a good way to approach it. Trying to think, what's our closest botanical garden?
Maybe a
community
Community college.
Yeah. Community college. I think we would be happy to host such an effort. And ideally that is what we're kind of slowly working on. It's tough sometimes. Maybe I should pause the recording before I say this, but you guys won't share it with. Anyway,
Sounds like you need a larger greenhouse.
yeah, we need a bigger greenhouse. I want to build another one myself, but yeah, I would suggest that sometimes we have competing views, right? And you know, some folks might want to see more of like the Disneyland model than what we think of as more like the rustic Appalachian model. But our area right here around the greenhouse, we're trying to slowly turn from invasive weeds that we currently have, especially the calorie pears that are kind of overgrowing our greenhouse.
You need a hypo patch.
Yeah, we need to get a hypo hatchet for that. That's but we were actually out there cutting some of them down today, and it's like removing that, but then putting something in its place. And I find that's something maybe lacking in a lot of invasive plant control efforts, is that you're opening up a niche, right? And if you don't get something back in that niche, it will fill back up with an invasive plant. So yes, and yeah, I'll get the hypo hatchet. If I can ever find it, it's just not.
I'll send you the link.
Is it in stock on there?
Yes. Who plants for
These are the
landscaping
pop-up
around
ads that stay
parking
in,
areas?
it's just part of the algorithm.
the main parking
He probably
areas around this house?
Oh.
A lot of grass.
Oh, yeah.
And there's no walkways. I mean,
Right.
you're always running into cars if you want to walk.
Oh, yeah. So that's another thing that we've been hoping to develop. We did get about a little over $200,000 to develop a arboretum on campus. So that's we think of doing it as a linear arboretum. So it would be a trail. So you know, we're years away from that happening, but that is something we're working on. So.
Is there any way to encourage it?
Oh, sure. Yeah, I think probably the first step that we're going to take in that direction is to kind of have like a public use survey. I'll be sure that you guys get that and say, yes, we want to see a trail on your campus. We have about 120 acres of campus here, and it connects to the urban pathway. If you could connect to the muster grounds area,
Yeah.
and then from there connects to the creeper trail to the AT to, well, you know how that goes. So connectivity, like that's another piece, and trails,
Yeah.
excellent place to you know, preserve and display native species of plants. And asexual propagation is definitely one of the techniques that we can use to get those plants in an affordable manner, right? So I think one of our issues, I'm sure we've all felt that heartburn, right? When we can find native plants, gosh, they're expensive. So we'll have you know, we're kind of limited currently, but we'll have about a bench of native plants available at our plant sales in April and May. So I would also encourage, and I'll try to get that message out when we have our sale dates advertised, but we will have some of the native plant material that students have been saving seeds from, and also asexually propagating, including some scarlet bee balm and some Virginia spiderwort. So the plants you see before you could be for sale. And we're cheap, you know. Other questions, comments, concerns?
When you were talking about the willow tea, Ben, I was just wondering, is that the whole Salicaceae family?
I think it is, yeah.
Yeah, so poplars would be.
You know, I think so, because I think it's maybe a precursor or of salicylic acid, or
Okay, yeah.
I'm not exactly sure how that chemical, what it is involved with that family, but yeah, I mean, all willows have it, so I'm sure that it is coming from, you know, maybe aspen as well, and probably some of the other poplars.
Or parts.
Usually the stems, so and probably the younger stems. So that's probably where the highest concentrations are, because we think of that as the in the growing point where you have the meristem is kind of where most of those hormones are stored. Yeah. Good stuff, though. I think it's really neat to, you know, I mean, it's great to use the hormone in the powdered form, but if you could get it from a willow and not have to buy it, 'cause it is expensive. That gel is like it's like $100 a quart. So yeah, it's costly, but I mean, a quart could go a long ways.
But can you divide that and put it in smaller containers?
Yeah, absolutely. Yeah, and then resell.
How did you say to make tea?
It's simple. Just take the branches, young branches of willow, stick them in a vase, and then it leaches out, and then you got it. So, and then I mean, some people actually take that willow tea and feed it to their seedlings to encourage root development too. So I don't know how effective that is, but sounds like an experiment we need to make. Other thoughts, questions, concerns? Not seeing anything. You guys out there in Zoom land are quiet. Feel free to unmute yourself and speak up or drop something in the chat. All right, well, there's no other questions. We'll go ahead and wrap up and invite anybody, you know, if you're zooming in tonight, you're always welcome to get in touch with me, and love to show you around what we got growing on here in our greenhouse and things that we're working on. And for those of you that are here, if you want to stick around for a minute, happy to show you some things in the greenhouse. And if you want to walk away with some tubers of some Jerusalem artichoke, I've got a ton of them.
She's gonna sit down here and salivating.
Thank you all. Happy spring.
Thank you.
Thanks, Ben.